Hi Bio30. In this video we're going to be talking about DNA, and we're also going to be going through the very first process that the cell goes through before it divides, called the interphase. So just to review some concepts that you guys would have learned in Bio 20, okay, talking about the chemical codes of cells. So nucleic acids, which are like the fancy name for DNA, are biological chemicals that direct growth and development of every single-celled and multi-celled organism by means of a chemical code. These chemicals determine how the cells function and what characteristics it has. So these are basically determining the sequence of DNA that an organism has. Depending on the sequence of letters in their DNA, you can have A's and C's and T's and G's. Depending on the combination of those letters, determines what an organism looks like, what type of proteins it can make, okay, all of that fun stuff. We have two different types of nucleic acids. So you can either have RNA, which is ribonucleic acid, which is shown here, which is a single strand. Instead of having A's, T's, G's, and C's, it has A's, C's, G's, and U's. Okay, we're going to see that this is kind of a messenger DNA, where it gets taken from the nucleus of a cell and gets taken to the ribosomes in order to create proteins. We're going to talk more about that in chapter 18. And then DNA is the main one, okay, that's in the nucleus of the cell that basically codes for different proteins and is the chemical instructions for the cell. Okay, remember that DNA is the molecule that gets passed on from parents to children. Okay, and basically each DNA molecule is instructions on making RNA, which eventually can make proteins like hormones and enzymes, things like that. Okay, and proteins are basically what cells are made up of and what controls functions of the cell. Okay, when you think about how many different things hormones influence and enzymes influence, okay, those are all proteins. But those also, just think about how many things in our body are made up of proteins, like connective tissue and muscle and the cells, okay, the proteins in the cell membrane that open and close, the channels that open and close and allow things in and out. Okay, there's so many different proteins, and how we actually make those proteins, we read the instructions in our DNA. So those combinations of letters tells us how to make those proteins. Again, we're going to talk more about that in chapter 18. This whole chapter basically focuses on cell division. So before we actually go through and talk about cell division, why the heck are we even bothering dividing? Why don't we just have one giant cell? Well, the reason for that is because if my cell is growing in size, my volume inside my cell is going to be increasing faster than the outside of the cell. So the surface area of the cell membrane, which is used to transport materials, nutrients into the cell and wastes out of the cell, isn't increasing as fast as the volume on the inside. So if that's happening, if we have an increase in volume, that means we need more materials transported because we have more cell that's needed to have nutrients and all that fun stuff. So if our cell is growing, okay, the membrane eventually becomes too small to support the cell within it. So eventually the cell gets too big and we'll have to divide into two smaller cells. Okay, if you continue to grow bigger, 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 bigger cells, the cell membrane is too small. Okay, so the surface area does not support the volume of the cell anymore. And also just think about how long it would take to get from this side of the cell to this side versus this side and this side. So it's going to take a lot faster to transport materials from one side to the other using diffusion or osmosis if the cell is a lot smaller. So that is why we have to go through cell division. We can't just have one giant cell. We have to be able to support all of the cell processes inside the cell using the cell membrane. The cell membrane has to be a certain size in order to support all the stuff that's happening on the inside. So when we're talking about cell division, we use the word cell cycle to describe one life cycle of one cell. So when it starts, okay, right after it divides into a new cell, that one cell, okay, is going to live for a certain length of time, known as the cell cycle, which is right when it's created to when it divides into two new cells. Then the cell cycle will start over with those two new cells. So depending on the type of cell we're talking about, depends on the length of this cell cycle. So if we're talking about something like a blood cell or skin cell, they have a very short cell cycle. It only takes them actually a few days to replicate into two new cells. But something like a nerve cell, their cell cycle is really, 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 really long. Or sometimes nerve cells don't ever recreate new cells. If they don't have a myelin sheath, okay, they're unmyelinated nerve cells, they would never, ever, ever finish the cell cycle. Once they are ruined, okay, or destroyed, or old, okay, they would just be gone forever. But skin cells and blood cells just keep replacing themselves over and over and over and over and over again. So when I'm talking about just one cell cycle, again, it's looking at this cell, it's brand new, it is growing larger, it's creating different things, it's doing its cell thing, and then eventually realizes it's getting too big and starts to go through the process of dividing, and then it divides into two new cells, then the cell cycle starts over. And it just keeps happening over and over and over and over again. So there are some important words that you're going to have to know for this chapter. So for example, when I'm talking about the original cell, I use the phrase parent cell. When we go through division, okay, we create two new cells. Those are called daughter cells. So our parent cell divides into two daughter cells. We also have chromosomes. Okay, chromosomes are condensed DNA. I think of them as little nuggets of DNA. So it takes that really, really, really long strand of DNA that can be, okay, meters or kilometers long, and it condenses it into little tiny fragments. Our DNA that we have in our cells is condensed into 46 little chromosomes. Okay, they can either be individual, which would be like a single line, or they can be replicated, which is like an X. Okay, and the replicated chromosomes are identical copies on both sides, so we refer to them, each side of them, as a sister chromatid. Okay, chromatin, on the other hand, is genetic material that is all unwound, so it's kind of like a big clump of DNA. And then a centromere on a chromosome that's replicated is the middle part that gets constricted or pinched in, okay, on a condensed chromosome. So this would be a chromosome, an individual chromosome. This is my replicated chromosome, so this is a sister chromatid. This is a sister chromatid. They're identical to each other. This middle part is a centromere connecting the two sister chromatids in a replicated chromosome. And then this, that you would see in the nucleus of the cell, most of the time, it would be chromatin. Okay, just kind of intertwined strands of DNA. Very, very long, very tangled. Okay, very hard to deal with. The reason that we condense into chromosomes is so that when we're dividing our cells and dividing our DNA, it's not getting tangled around each other. If we tried to separate our cells and we had chromatin to deal with, when we tried to separate our DNA, it would all get kind of tangled and mixed up, and it just would be a big mess. Okay, the next thing that we need to know is what a homologous chromosome is. So humans have 46 chromosomes, but they have 23 homologous pairs. Okay, you get 23 chromosomes from your mom and 23 from your dad. So basically all of these chromosomes can be paired up. Okay, chromosome 1, 2, 3, 4, 5, 6, all the way up to chromosome pair 23. On the 23rd pair is where you have XX or XY, which determines the sex. So if you're XX, that would be female. XY would be male. Okay, but every single homologous chromosome carries genes. They carry the same genes. They're also the same size, okay, the same shape, and they have the same banding pattern. So you see these dark kind of lines on the chromosomes. They match up with each other. So this is a picture that you would use to like see the matches of homologous chromosomes, but the reason that they match up is because they carry the same genes. So maybe chromosome 1 carries eye color, hair color, whether or not you can roll your tongue, but because your parents are different, you might have different versions of that gene. So if your mom has blue eyes and your dad has brown eyes, and you're inheriting brown eyes from your dad and blue eyes from your mom, okay, you might show yourself as having brown eyes, but you're carrying blue eyes. So on your homologous chromosomes, they carry the same genes, but they might look a little bit different. They might not be genetically identical because half of them are coming from your mom and half of them are coming from your dad, and I guarantee that you're going to have some differences between your mom's DNA and your dad's DNA that you're inheriting. So those are homologous chromosomes. And then genes are specific locations on chromosomes or on DNA. So you could see the gene for eye color could be right here and right here in both of these, but it's just a specific combination, okay, specific location of the DNA, and depending on the combination of your A's, your C's, your T's, and your G's, depends on what it would look like. So in order to have blue eyes, you have to have a certain combination on your eye color gene of those letters. In order to have brown eyes, it would have to be a different combination of those letters. And then we know these words from the last chapter, but diploid means 2N, which means you have 2 copies of each chromosome. You have pairs of homologous chromosomes. Half of that would be unpaired chromosomes. This is what we see in a sperm or an egg, a haploid cell. We only have half the amount. And then sometimes organisms are what we call polyploid, which means they have more than 2 copies of their homologous chromosomes. Okay, strawberries are 4N, which means that they actually get 2 copies of each chromosome from their mom and 2 copies from dad. So instead of having their picture look like this, okay, with 2 chromosomes, they would have 4 on each part. Okay, so that's what polyploid means. Underneath that table, you guys have a spot to draw a little diagram. So basically just wanted to show you all of the different types of versions of DNA, how they can look at different spots. So in the nucleus of the cell, usually the DNA will appear as chromatin, which is kind of a jumbled mess. Okay, if I looked at the individual strands that are in this chromatin and zoomed in, it would look like my double helix of DNA. And remember, specific pieces of this double helix of DNA are genes. They're always found in the same location. We've mapped out every single location of every gene in the human DNA. If I coil up my DNA into little nuggets, okay, depending on if it's individual or replicated, they're called chromosomes. And remember, sister chromatids are a replicated chromosome. Either side of them are identical to one another. So moving along to the cell cycle again, the cell cycle is just the cell as it grows and then eventually divides into 2 new cells. But it goes through 2 different phases within the cell cycle. So we have interphase, which is the majority of the time, which is what the cell would look like here. Okay? And then cell division is when it's actually separating. So going through the process of lining up the chromosomes, okay, separating the chromosomes, eventually forming 2 new cells. That would be cell division. Within interphase, there are 3 separate phases. So the first phase is G1, which is defined by growth. So basically the cell just doing normal cell things. So it's going to build new organelles, okay, produce lots of proteins depending on the cell. Okay, so if it's a pancreas cell, it's going to make a whole bunch of insulin and glucagon because those are the proteins that it's in charge of making. Depending on the cell, though, it makes different proteins. But basically just being a regular cell. Okay, before it gets to this next phase called S phase, it's going to get a signal saying, uh-oh, you're getting a little bit too big. So you have to start figuring out and getting prepared for that cell division. So in S phase, it's going to take the DNA, it's going to replicate it, so that when we separate the cells, they have identical copies of DNA. And then the last stage is just basically increasing energy in order to get through that division, and also just continuing making proteins and just mentally preparing for that division. So if I was going to look at a pie chart, okay, I would divide the pie chart into these divisions. So starting with this part right here all the way around here, so I would say like 95% of the cell cycle is called interphase, which is divided into 3 separate sections. And then this small little sliver is how long it takes for the cell to actually divide. So most of the time the cell is hanging out in interphase. It's a very, very small amount of time that it would be dividing. Okay, the different phases of interphase, we start with G1. This is the longest phase of interphase. Remember, this is where it's growing and it's doing metabolic activities, so basically just being a cell, making proteins, making organelles, okay, doing whatever its function is. Okay, then it hits this middle mark and it's like, oops, I'm getting a little bit too big, I gotta start getting ready for that division. So it goes through S phase, which is DNA replication, and then G2 is when it's basically doing that final preparation for division, making more energy, okay, continuing to make proteins, stuff like that. So that is the cell cycle, and that's actually the end of the video today. So you do have to know the cell cycle and be able to draw it out, but also answer some general questions about the stuff that I was talking about in the start of the video. So that would be your drawing quiz tomorrow, drawing out this picture right here, and then also answering some questions about the other information in the video. So hopefully you have a good night. I'll see you guys tomorrow.